So continuing on here, the Wright-Giemsa stain, that is that sequence of the 3 staining bottles, with the first one being a fixative, and then the second bottle being the orange eosin dye, and then the last one being that purple basic dye, it's crystal violet. And you're using that to distinguish mostly the basophils from the eosinophils, because the other white blood cells you can distinguish by the shape and form of their nucleus. Now the basophil and the eosinophil, they look alike, and if you don't distinguish them with the eosin versus crystal violet, then you would not be able to tell which one is which. Anyway, so that's on 8. Number 9, what problems may occur making differential white blood cell slide preparations difficult to interpret. The biggest problem is that the smear is too thick. Now in the Labster exercise, they basically just showing you how to do a smear. It has to be a really thin film. It's nice, it would have been nice to practice that, but it is what it is. So just take a look at the Labster exercise, and then also they're showing you a screen, and then you can take a look at the different white blood cells and also different shapes of red blood cells. They're showing you sickle cell anemia, and also they're showing you somebody with malaria, which is kind of interesting. On 10, outline the major visible differences between granulocytes and agranulocytes. So on the granulocytes, real quick again, the neutrophils, the one with the polymorphonuclear shape there on the nucleus, so many shapes on the nucleus, a segmented nucleus, and then the basophil and the eosinophil, you can only distinguish them by their staining properties of their granules. On the agranulocytes, that's pretty easy because the lymphocyte is relatively small with that huge nucleus inside, and the monocyte is a big cell with a kidney-shaped nucleus inside. So all of them are really easy to spot on a microscope slide, and just go ahead and do that Labster exercise to show you what they look like. And we'll also talk about that in lecture when we do the blood lecture. And number 11, neutrophils. Cytoplasmic granules are generally not visible with a Wright-Giemsa stain. Explain. Well, neutrophils, they like a neutral dye. We don't offer them one, so they don't like either the eosin nor the basic dye, because they're neutrophils, and therefore they don't stain with either orange or the crystal violet, that purple dye. So, but we don't need them to stain because the shape of the nucleus is so characteristic that neutrophils are just easy to spot. Number 12, can a differential white blood cell count to be used to distinguish T cell from B cells? You can't, because those are the different types of lymphocytes, and you would not be able to to distinguish whether that is a B cell that makes antibodies or a T cell that engages in cell-mediated immunity. So the short answer is no on 12. 13, compare the average hematocrit for males and females in the class. Well, we can't really do that, but on average, the hematocrit is in fact higher. And if we were to do a t-test, like we are asked to do on 14, we would find that there is a significant difference between male and female hematocrit. And number 15, would you expect the hematocrit measurement to be high or low? Well, there's some evidence that nicotine might increase your hematocrit. So some even say that it might lead to polycythemia. There is some controversy around that, so I'm not quite sure what to make of the data there. But some people say that smokers tend to have a higher hematocrit. High altitude, definitely. So lower oxygen levels will trigger the kidneys to release erythropoietin, and erythropoietin will then direct the bone marrow to make more red blood cells. So yes, you will have a higher hematocrit, and it can be quite dramatic actually if you go into extreme altitude. Let's say you wanted to climb Mount Everest. First of all, you have to adjust to the altitude gradually, and also if you then go into these extreme altitudes, like 29,000 feet or something like that, up high altitude, then the hematocrit can get so high that the viscosity of your blood is so high that your blood almost gels in your cardiovascular system, and it causes cloggages and altitude embolism, and it can be deadly. So this is quite dangerous because your body just basically reacts and tries to compensate for this lack of oxygen or this lowered amount of oxygen, and too much erythropoietin will lead to these ultra high hematocrits, and that can be quite dangerous. Low altitude, on the other hand, unless you go really far below sea level, that shouldn't really have much of an effect, unless, who knows, if you go really low altitude, but that's not common. And then polycythemia, that means you have too many red blood cells, and that means your hematocrit would then be accordingly higher. On 16, what is the importance of measuring plasma protein concentration? Well, the big issue here is the colloid osmotic pressure. I'm going to write it down. And that's a really important concept, in my opinion, because you need to understand that capillary beds are always leaky, and so there's going to be a lot of water that's going to leave in a capillary bed. Now, if you don't want to have edema, then you need to somehow recapture these fluids that have left in a capillary bed, and so you need to have a significant colloid osmotic pressure to pull the water back into your cardiovascular system. And if your colloid osmotic pressure would ever be too low, let's say you have renal failure or your glomerulonephritis, and now you are peeing out your plasma proteins, and now your plasma protein concentration keeps getting lower and lower, then you would end up with edema. So first thing, if I would see a patient coming in the hospital with edema, first thing I would check is how are the kidneys operating. Second place I would check would be the liver, because the liver makes those plasma proteins. But anyway, if a patient pees out the plasma proteins, you know that the reason for the edema is that the kidneys are not operating correctly, and they're allowing the plasma proteins to get out, and so then you're peeing them out. Now, here's another thing. If you are starving and your body doesn't have any kind of nutrition to make energy, so no carbs, no fats coming along, somebody's starving. You see this in kids that are starving. They have these hunger bellies. Now, obviously, that's not a big belly from too much food, but it's ascites, it's tissue fluids, because their bodies have started to digest the plasma proteins, and so now their colloid osmotic pressure is too low to recapture fluids, and so now the fluids start to accumulate in their bellies, and then they have these ascites. It's called a hunger belly. So something to consider. I find that whole concept of the plasma protein concentration and the colloid osmotic pressure is very important for physiology students to understand. Okay, moving on. Compare your hemoglobin concentration with that considered normal for your gender. Well, in general, women have, on average, a lower hemoglobin concentration than men, and we just can't compare because we didn't do it in the lab, the class data. And so also we're going to skip over this t-test, and it would show that there is a significant difference between male and female hemoglobin concentration. And then the percent ABO blood types, it usually follows pretty closely the pattern that was listed in the lab manual, with the majority of people having blood type O, followed by A, and then B, and then a few people with AB blood type. The RH level also usually follow the typical pattern, with the vast majority of people having being RH positive, and so the lab manual and just about any textbook will tell you that about 85% of the population is Rh positive and about 15% Rh negative, and we usually see that in our labs as well. Now, the last question: explain why an Rh negative woman should be concerned if she's pregnant with an Rh positive fetus. So again, the whole issue is this erythroblastosis fetalis. And what happens is that if a woman is Rh negative, so she's Rh negative, she does not have this antigen. If she's pregnant with an Rh positive fetus, then since the blood supply never mixed between mother and child, it's not a problem at first. But toward the end of pregnancy, latest at birth, the mother will be exposed to her baby's blood, that Rh positive blood. And her immune system will right away recognize this as a foreign antigen and will start making antibodies against the Rh factor. That's not going to affect the first child, but if she gets pregnant again with an Rh positive fetus, then she already has the antibodies. They will cross the placenta, and as soon as the baby starts making its own blood, then these red blood cells will be attacked by those antibodies. They will agglutinate and they will kill the fetus. Those were women in the times before 1950s, when this was figured out. Those were women that had one child, and every subsequent pregnancy would end up in miscarriage. Around 1950s, people figured this out, and a shot became available that absorbs any antibodies, these Rh antibodies that a mother might make. That's called the RhoGAM shot. And a woman, an Rh negative woman that's pregnant with an Rh positive baby, will get that RhoGAM shot to prevent the erythroblastosis fetalis. Let me write that down. Okay, so here's your term one more time: erythroblastosis fetalis. And that concludes our hematology lab. I highly recommend the 2 lab star exercises. I think they're quite good. And just have fun with the hematology lab. And make sure that you study the important terms, that you know about blood typing and all the other terms that are on the front page, because you will have a quiz on this.